Hey there, welcome back. Get ready for the final video in this series on molecular and cellular vocabulary A through Z. Today we've got a lineup of terms from U to Z. Stay tuned. Welcome to Science is You, a new channel dedicated to making real biotechnology research enjoyable and accessible to everyone. To support this mission, please subscribe and spread the word. Thanks for watching. U. Ubiquitin. Ubiquitin is a small protein found in many organisms, including humans, plants, and yeast. It plays a role in lots of cellular functions, including cell division, regulating proteins, and DNA repair. V. Vesicle. A vesicle is a small, bubble-like cellular container used for transporting fluids, gases, proteins, and other molecules within or outside cells. It can also be used to transport items between cells. These extracellular vesicles can play important roles in cellular communication and transport molecules that regulate protein production and alter cellular behavior. Vesicles come in a variety of sizes, much like Tupperware, and can play many and varied roles in transport of cellular items. They can be used to take in nutrients from the surroundings and dispose of cellular waste. W. Wild type. In biology research, organisms like cells or mice are often studied under different conditions. For example, a mouse with a mutation that causes tumors to form, or cells that have an extra copy of a gene. But if we want to see how these organisms respond differently to their environment or to potential medications, we need to compare them to something that doesn't have that mutation, genetic change, or other feature. This experimental control is often called wild type. Think of it as what occurs naturally in the wild. But it could also just mean the standard, unchanged version of a laboratory organism. X. X-ray crystallography. X-ray crystallography is a method of determining the structure of proteins or other large molecules. Most proteins are generally too small to see their intricate structures with even our best microscopes. To get around this, researchers back in the early 1900s took advantage of the observation that light could be spread out in predictable patterns by crystals. Crystals are a state where molecules are arranged in an organized, repetitive pattern. When light waves encounter the edges of molecules in a crystal, they spread out and recombine, making patterns of light after passing through the crystal. But to study protein structure, light waves are needed that are much smaller. X-rays, although not visible light, are a type of electromagnetic radiation, i.e. light waves, that have high energy and tiny wavelengths compared to atoms. By examining the patterns created from beaming X-rays through crystallized solutions, we can infer the structure of the proteins or other molecules. For example, this technique was instrumental in the discovery of the DNA double helix structure back in the 1950s. Y. Yamanaka factors. Embryonic stem cells are valued in biomedical research for their ability to turn into practically any type of cell in the body, a state called pluripotent. If this ability could be harnessed, we could perhaps replace organs or cell types that have become damaged due to injury or degenerative disease, or find cures for genetic diseases that cause types of cells to malfunction. However, embryonic stem cells also have a number of issues, including ethical concerns and potential rejection or attack by the immune system. Wouldn't it be great if we could just take cells from, say, your skin and make stem cells that are unique and matched to you? A discovery made within the past couple of decades showed that it is indeed possible to take adult cells and reprogram them into cells with properties very similar to embryonic stem cells, called induced pluripotent stem cells, or iPSCs, by inserting active copies of 4 genes that are critical for stem cell function. These genes were named Yamanaka factors after one of its discoverers. Z. zDNA. DNA is a very long, winding molecule that can take on different shapes and structures. DNA structure plays important roles in what protein machinery can attach to it where, which regulates whether genes get activated and copied for the production of other proteins or not. The most well-known structure is the classic double helix, or B form. This famous structure is formed by 2 strands of DNA that form a right-handed spiral, or helix, that moves clockwise away from you if you're looking at it from the top. But DNA can also spiral in the other direction. This left-handed helix is called zDNA. This alternative structure has biological roles, including immune response, and is under research as a potential target in immunotherapy approaches to treat cancer. There you have it, terms U through Z and the final video in this series. What did you think of this series on molecular and cellular biology terms A through Z? Share in the comments. If you enjoyed this video, please subscribe and remember,